All right, so for the first topic that we'll be going over, cell replication, you'll find this in your textbook on pages 352 to 356. Now, your textbook, it talks about the purposes of cell replication, and it talks about the different kinds of cell replication that you might observe in prokaryotes and in eukaryotes. And there is a difference, and you do need to know this difference at the VCE level. So in this video, we'll go over all of this again. So starting off with identifying those purposes of cell replication. Now, there are three major purposes that the textbook gives for cell replication. The first of these is for growth and development. So the first of these is growth and development. So organisms typically need to get bigger. So once you were a little embryo and you slowly grew into the person that you are nowadays, and you might be growing still, you might continue to get larger. And this happens, this increase in size happens as a result of cell replication. So that's one of the reasons. Now, the second reason is for cells to restore their nucleus, to cytoplasm ratio. Now, what does this mean? So the nucleus is the control center of the cell, and from the nucleus, we have all the cell's instructions for the production of proteins and other functional units in the cell. Now, if the nucleus is there's only one nucleus in every cell, and the cytoplasm can vary in size. So if the cytoplasm, which is where all the stuff, all the cellular processes happen, if the cytoplasm gets large and too large, the nucleus cannot any longer effectively manage the entire cellular space. And so one way that cells combat this problem is to split themselves. And when they replicate and split themselves, they reduce the size of the cytoplasms, and that makes it easier for the nucleus to control what happens in the cell. Okay, so that's the second reason, and the last one is for maintenance and repair. So cells often accumulate damage over time, and it becomes important that they replicate themselves into new cells so that the damage can be removed by destruction of the old cell. So a new cell, which is an old cell, which is like an old broken cell, can replicate. And when it replicates, it makes a new fresh cell. And the old broken cell is still there as well. So there's two of these cells now. There's the old broken cell and then the new fresh cell. And the old broken cell now can be eliminated by apoptosis, so programmed cell death. And we're left with a new fresh cell which doesn't have all the damage of that older broken cell. Okay, so we've got those three different purposes for cell replication. So whenever you get a question which asks about why we need cell replication, at least in eukaryotes, so multicellular organisms, we have these three reasons. Okay, so now prokaryotes and eukaryotes. Prokaryotes and eukaryotes. So cell replication in prokaryotes and eukaryotes. Prokaryotes and eukaryotes have different ways of replicating. With prokaryotes, they undergo a process that we call binary fission. And in eukaryotes, we have mitosis. So you need to know these two different processes and exactly what kind of cells use which process. So with binary fission, and I'm just going to rip this this diagram out of the textbook with binary fission. I really want to, with binary fission here. I already got it. And paste. Nope. Hang on, I'll just get this copied over. Okay. Binary fission can be visually represented like in this way. And so we know prokaryotes typically have circular DNA. They don't have their DNA on a number of linear chromosomes like in eukaryotes. They have them in circular chromosomes. And so because they don't have any like organelles and the membranes that need to be broken down in organelles and so on, it's really simple for them to replicate this DNA and then just split it into two new cells. So you have this DNA here that it sits, just floats around in the cell, and at some point starts to get replicated. So here we have replication, DNA replication beginning. And then it continues until you get two identical sets of circular DNA. Two identical chromosomes. And once we have two identical chromosomes, these are going to be divided into or separated into two different parts of the cell, which then starts to divide. So a cleavage furrow here forms. This is what they call it, a cleavage furrow, and then this splits the original cell into two new cells, with each of the two new cells having the same set of DNA, so the same chromosome, which has just been replicated. Okay, so this is binary fission. Now mitosis is a lot more complex than binary fission, and the reason why it's more complex, the reason why we find mitosis to be more complex is for a couple of reasons. First, we have DNA being inside a membrane-bound organelle. So that means the DNA is like wrapped inside a plasma membrane. So this makes it a little bit more complex in terms of how do you release that DNA from the membrane-bound organelle and then divide it into two new cells and then reform that membrane-bound organelle into two new cells, because DNA must be bound within a plasma membrane. So that's one of the issues. And the other one is that we have multiple chromosomes in eukaryotes. And because you have multiple chromosomes within eukaryotes, there has to be a way that we effectively replicate all of these chromosomes and then divide them perfectly into the two new cells. So they need to be replicated, each chromosome, each must be replicated perfectly, and then each new cell needs all the different chromosomes. So all of the different chromosomes, no more, no less. So once a cell divides, it needs to have, in humans, 46 chromosomes, 2 of each type. So if you have more than 46, you've got a problem. Less than 46, you also have a big problem. So it's really important that these are achieved in mitosis. So it's a lot more complicated than binary fission. In terms of mitosis, we'll look at mitosis on another page, but just generally what was required here. I think we just name it here. So we just say mitosis, generally speaking. It's made up of 4 steps. We just say PMAT. But mitosis is part of what we call the cell cycle. So I'll put that into part of the cell cycle, which is a very particular process by which cells prepare for replication. All right. So we've looked at the purposes of cell replication, and hopefully from what was discussed here, you can distinguish between binary fission and mitosis in terms of the complexity of those 2 processes. I think it's important that you can describe generally how binary fission occurs. So if you can just step through the process of binary fission, I think once you know in more detail, you should know in more detail the process of mitosis, it should be easy to distinguish between the 2. And yeah, so here we have a little discussion about why mitosis is needed for eukaryotes. So this is a result of there being the DNA being bound within plasma membranes, and also the fact that there are multiple chromosomes.